City. So I will share my presentation. So my presentation is on recent advancement in the development of microbial biotherapeutics in the battle of antibiotic resistance. So actually, we are in a platform in which we are collaborating with multidisciplinary aspects, but there is one important point, that is awareness. So this slide, my slide is a bit of what I do in my research with the students and all my research papers which have been published. So it is basically a consortium of all those things. So this is one of the most important global threat is antibiotic resistance. If I move on to the introduction, that antibiotic resistance basically stems from overprescription and also by irrational use of antibiotics. If I go back to 1928, we have all heard about Dr. Alexander Fleming. So he has changed the era of disease, the chemotherapy, that is, he has found out by his accidental discovery, penicillin. But during his receiving of Nobel Prize, along with Ernest Fleury and Chain, he said one thing, that if human beings irrationally use antibiotic, if they do not take right dose, one day it will come that there will be no antibiotics to treat microorganisms, to treat bacteria, to treat disease. And that is the present problem of our society. So if I move on to my next slide, I will give you the global statistics that you see that over 7 lakh deaths annually due to antibiotic-resistant infection at Lancet. According to Lancet, by 2050, it could cause 10 million deaths per year if not addressed. And the economic impact, that is antibiotic resistance, could cause a global economy of $100 trillion by 2050. And the major regions which are affected today is Asia, Africa, and Europe. So I have also taken data from the global statistics of antibiotic resistance and have found out that in Asia, due to high rate of overuse of antibiotic in human and agriculture, that is the main cause. In Africa, limited access to quality antibiotics. In Europe, increasing resistance to last line of antibiotics like carbapenem. If you go into the hospital, you will get to see that most of the patients who are there in the ICU and they're suffering from pneumonia, the first line of defense they are given is carbapenems, meropenems. And these are the antibiotics which save these individuals. But these, unfortunately, even the microorganism has gained resistance. Again, this line of antibiotics which are used for nosocomial infection and even infection acquired in the hospitals. And also not in North America, raising rate of drug resistance infection in healthcare settings. So there is an urgent need of alternative therapeutics. So for alternative therapeutics, actually, we are working on few therapeutic approaches. Here, I want to say that a bacteria, a microorganism will kill a microorganism. That is my approach. That it is the microorganism which is killing us, but it is some of the newer therapeutics isolated from microorganism, from bacteria, viruses, which will kill another bacteria which is resistant to multiple different kind of antibiotics. And that is called as antibiotic resistance. And even we also have multi-drug resistance. So the first I have here in my presentation, I have given a brief about bacteriophage therapeutics. Antimicrobial peptides, we are trying to isolate proteins and peptides from various kinds of bacteria, and we are trying to apply it on as an antibacterial agent on drug-resistant pathogens in our laboratory. And we are also focusing on another important aspect, which is aggravating the drug resistance, that is antibiofilm, that is they are producing biofilm. I think all of you have heard about that whenever sometimes there is an operative failure of knee replacement. That is because of the growth of microorganism in the implant, and that is because of biofilm. So if an antibiotic resistant bacteria also have the ability to produce biofilm, it is very, very difficult to combat. And there is another important thing, that is your postbiotics. All of us takes prebiotic, like we have all heard about lactobacillus, we have all heard about curd, in which we get good bacteria. Even when we go to the doctor, if we take an antibiotic for long term, they give you probiotics, but there are so many different metabolites which can be isolated from this organism and can be utilized for antibiotic resistance. So that is basically my work. So this is my first work, that is bacteriophage therapeutics, and this work is having a huge, they are in AIMS, All India Institute of Medical Science also, a huge amount of money has been mobilized, a lot of grants are being given for this particular work. This is actually a virus which is infecting a bacteria, which is killing a bacteria. How? This virus are entering into the bacteria, multiplying in the bacteria, lysing the bacteria and moving on to another bacteria and getting reinfecting a healthy bacteria. So what is the main thing? Why it is so important? Because these viruses are very specific to a bacteria. It doesn't affect all other cells. Like if I talk of antibiotics, it can also affect some non-specific cells. But this doesn't it doesn't infect any non-specific cell that's specific to specific bacterium. So we are trying to isolate this kind of bacteriophages from sewage water, and we are trying to test against, we're trying to grow against this sensitive organism, which are basically MDR strains we are having, like MRSA, Pseudomonas aeruginosa, Staphylococcus saprophyticus. These are some of the pathogens of clinical significance. And we are amplifying those phages, those viruses, purifying them, characterizing them, and also formulating a delivery system, that how we can deliver this particular phage such that it can be targeted to the correct tissue where this organism, this bacteria has invaded. So these are certain studies on immobilization of bacteriophages on various surface in order to develop phage formulation. So we are focusing on 4 techniques like physical adsorption, protein ligand, electrostatic interaction, covalent linkages, and we are trying to, we are isolating phages. There are many other phages. These are some of the review work which we have done for starting our projects. So we have got this idea of immobilization of phages, and ultimately we have come up to various phage formulations. So there are various phage formulation till date, and people are working on it. That is, we can go for a phage cocktail where multiple number of phages can be utilized at a time against various microorganisms. Even we can isolate that particular gene from the phage, and we can use recombinant DNA technology to proliferate enzymes which can kill microorganisms, which can kill bacteria. We can engineer phages. We can also, combine the most innovative technique, that is CRISPR-Cas, with the phage, as well as one of the easiest way what we can do is that if we can establish a phage, we can also combine various type of antibiotic to find out that if there is any kind of synergistic effect of antibiotics and phages. So that is basically one part of the therapeutic approach we are taking against antibiotic resistance. Another is bacterial proteins and peptides. So we are taking bacteria of risk group 1. Maybe we are taking bacteria of risk group 1. Why? Because risk group 1 are those pathogens which do not cause any kind of disease, and we can easily handle it. And we are producing this bacterial biomass to isolate the intracellular proteins from them and screening those protein pools against test bacterium, which I have already talked about. And then we are trying to purify the protein. We are trying to purify that protein pool and subjecting it to STS, polyacrylamide gel electrophoresis for protein separation. And then we have seen the overexpression of the protein, and accordingly we have separated by gel permeation chromatography. And we have taken the fractions and we have studied on each fraction, followed by characterization of this protein by mass spectrometry CD, or also we have used MALDI to determine this bacterial protein or characterization of this protein. So this bacterial proteins can also be an alternative approach, though we need with protein stability is a very, very important factor, and how we can deliver those protein. The formulation is also another important factor which has to be taken into account after establishment of the proteins and peptides. So here comes the lactobacillus, which I am talking about, that is their secondary metabolites or postbiotics. So what is happening is that when in fermentation, when I'm growing a microorganism in a stress condition, what they do is that in a stress condition, they produce growth inhibiting substances. That is basically called as the secondary metabolites. And those who are producing this growth inhibiting substances, they are immune to it. While those who cannot produce, they are getting killed. So that is the basic principle of this postbiotics, which we are trying to isolate from these microorganisms. And we are targeting lactobacillus species. These are already work done. You can see the references also that Listeria monocytogenes was targeted upon by Lactobacillus plantarum J23, E. italicus ONU 547, and so there are hundreds of bacterial species of lactobacillus from where these proteins, postbiotics are basically being isolated and targeted upon various bacteria. Even Salmonella typhimurium is also being targeted. So these, we are basically also converting this different antimicrobial peptides into various types of like nanofibers, which is also called as nanobiotics. And also we are trying to formulate, like we have conjugated bacteriocin nanoliposomes, bacteriocin reverse myceli. So these are basically our approaches for targeting various microorganisms. So this was another aspect of my presentation, though I will just conclude it. So that is our biofilm inhibition, which we have done with bacterial proteins and biosurfactants. So some works are there on it as well. So we are trying to target the EPS generation, exopolysaccharide generation. We have also found out, we are working on quorum sensing quenchers, which can prevent the biofilm formation. And finally, so these are some of the review we have done. And these are the classification of biosurfactants which can be utilized, which has already been utilized and which can be utilized for multi-drug resistant bacteria. Though these bacteria are not multi-drug resistant, they are the sensitive bacterium, but they can be utilized. There is a huge prospect of using this biosurfactant on drug resistant bacteria. And this was a small online survey which we have done to know the awareness of this microbial biotherapy among our students. So the awareness is very less, as we can see from the results. So I will conclude by the point that in future, we can have various types of approach which can be combined together to target antimicrobial resistance, which is a global threat and which can cause a huge amount of problem in near future. So these are some of my papers which I have published, that is on bacteriophage therapeutics. These are some of the papers I have published on antimicrobial proteins and peptides. And even I have worked on silver nanoparticles and their antibiofilm activity on Staphylococcus saprophyticus. And also these are some of my papers where I have worked on secondary metabolites and their antibiofilm property, and also some contribution of antibiofilm property in antibiotic resistance. With this, I want to thank the organizer of this conference for giving me the opportunity to explain what work we are doing. Though I have not given the data, but these are some of the works which we are doing in our laboratory.